And thanks to Lara and the whole ICGEB for inviting me to talk. I always am happy to talk. In my work, our work with CD101 has been an exciting um, process of discovery. So I'm hoping I can get all of you excited about it too. So just to start out, let me see. I seem to there. Um, so the outline of this talk, I'm just going to start first with a brief discussion about treatment and prevention context of HIV-1 um, and why that status of treatment and prevention it motivates us to continue to have an interest in why some people are highly HIV who are highly HIV exposed remain HIV seronegative without prevention interventions. Um, and then I'll move on to talk about the study design that I started with, namely the extreme phenotype study design, and how we use that to identify CD101 variants as being involved in altering HIV acquisition risk. I'll then go through a series of collaborative studies that I've been involved with to understand the mechanisms underlying CD101's association with HIV, and then highlight some additional recently published findings from other groups that have demonstrated the growing impact or understanding of the impact of CD101 on inflammation and infection in general. And my major take-home points for this talk will be that the extreme phenotype study design can be used to identify genetic variants with large effects on disease causation. And the genetic variants in the gene CD101 can strongly increase the risk of HIV acquisition, and that this increased risk may be due to CD101 variants causing dysfunction in innate immune cells that lead to higher numbers of HIV susceptible activated T cells. Um, and so let me start right off with this background of the treatment and prevention landscape of HIV-1. Clearly, we started in the 1980s with HIV being essentially a death sentence. The infection with HIV, we had nothing to intervene with. CD4 cells, T cells were hammered by the virus, and over the period of months to years, the degradation of the immune response led to the vast majority of people dying from the infection. And transitioned to 20 years, 15 to 20 years later, with the development of combination antiretroviral therapy, the um, the impact of HIV, while still serious, the people who are living with HIV could continue living with relatively normal lives as long as they were able to maintain their antiretroviral therapy, where CD4 counts, T cell counts came back to normal and HIV RNA was undetectable. Um, that development of antiretroviral therapy then transitioned to also being useful. Some of those same drugs were useful in antiretroviral chemoprophylaxis, PrEP, which was instrumental in providing a preventive measure, a chemotherapeutic or chemoprophylactic preventive measure for HIV. Um, which then broadened the public health impact of these interventions. The disappointing part of HIV prevention has been the results of efforts to develop HIV vaccines. And as this table shows, for the last over 25 years, there have been numerous efforts to use a variety of approaches to develop HIV vaccine, an effective HIV vaccine. And of these major efforts, one showed some sign of efficacy. The RB144 Tide trial showed some efficacy, but efforts to try to replicate that or enhance that efficacy were met with failure, and we still find ourselves 40 years into the epidemic with still no effective HIV vaccine. So this lack of preventive, immunologically preventive intervention is motivation to better understand why some people remain HIV seronegative despite high HIV exposure. Um, and the first example of a host factor that was documented as associated with HIV, with innate differences in HIV susceptibility, was the genetic variant CCR5 delta 32. Back in the late 90s, this deletion variant in the chemokine receptor CCR5, which is a co-receptor for HIV virus, was identified through studies of cohorts that had high HIV exposure through blood transfusion or blood product transfusions or multiple sex partners. Um, and it was identified that this variant, which is present in about 10 to 15% of some Northern European populations, and not present in other populations, including Africans, um, was shown um, to result in resistance to HIV infection. And this actually led to interventions that included development of the HIV entry inhibitor drug Maraviroc, which is used in a variety of contexts clinically today, um, but also this knowledge was used to develop interventions, including bone marrow transplant, that have been able to affect HIV cures, but only in a handful of individuals, given the cost and the intense efforts needed to implement these type of bone marrow transplant efforts. But it at least in theory presented an opportunity to cure HIV. Um, but in some ways, CCR5 delta 32 is more of a block to virus replication and less of a change to the immune response. And so the question remained, are there factors that alter HIV susceptibility through innate changes in the immune system? And we could get some support for the idea that this was still plausible through cohorts that have been identified from following people who were exposed to HIV. An example of this was the cohort of exposed HIV exposed seronegative sex workers in Kenya, in Nairobi, Kenya, that has been followed. This cohort has been followed from the late 1980s through the early 2000s. And this graph of the probability of remaining seronegative for this cohort shows that the data, the survival analysis curve shown in the black dots. I don't know if you can see my pointer that. So these black dots show the data, the actual HIV survival or HIV infection data of the cohort. That survival curve has a better fit to a model in the dark line here that includes the cohort having a subset of the population that is resistant to HIV. So the model that best describes the survival data is one that incorporates a population that's resistant to infection. So the question from cohorts like this remains that what are the mechanisms that differentiate people who remain seronegative, these groups, this group here, from the people who don't remain seronegative over time in a cohort such as this. And extensive immunologic studies of this cohort have still not clarified the mechanism that's involved in protecting these particular individuals. But these immunologic studies did lead to the idea of immune quiescence, the idea that something in the system is quieting the system's responsiveness, and thereby making it less likely to get infected by HIV. But what that mechanism is and how we can use that to protect other individuals has still yet to be fully explained. Um, Recognizing that cohorts like the Nairobi sex worker cohort did demonstrate some mechanisms for host protection against HIV, we at the University of Washington had been actively involved in coordinating cohorts of HIV serodifferent heterosexual couples, and we thought that this would be a unique context in which to look at that same question of protection from HIV or susceptibility to HIV. And so this study design is where we follow up on stable heterosexual couples with one partner living with HIV and their regular sexual partner living without HIV at enrollment. And then we follow them over time, collecting data and samples from both partners to quantify factors associated with the risk of acquiring HIV. Um, Over the last 25 years, we have coordinated two large clinical trials and two large observational studies that have recruited about 9,000 heterosexual couples, of which 300 were identified as having experienced an HIV seroconversion event, with up to 3 years of follow-up data on each couple. We've confirmed the HIV transmission linkage between the two partners whenever there is a seroconversion through sequencing the plasma virus from both partners in those seroconverting couples. And we've also included consent for genomic studies in a subset of the sites. So this seemed a unique opportunity to look at HIV susceptibility and resistance in the context of these serodifferent heterosexual couples cohorts. And so um, let me go into the first discussion of our collaborations to understand this, the biological factors or identify biological factors associated with resistance or susceptibility to HIV. This is a collaboration. The complete genome sequencing project was a collaboration with Dr. Rommel McElprang, who at the time was a postdoc working with me, Dr. Mary Emond, a genetic statistician, um, and Dr. Michael Bamshad, both at the University of Washington. Dr. Bamshad is a genomic scientist here at the UW. And so this team um, took the approach of saying, um, what are the genetic factors that differentiate people who become infected versus people who don't become infected with HIV? Um, and we couldn't simply compare the genomes of people living with HIV versus people who were living without HIV, because we knew that HIV infection is altered by epidemiologic exposure factors, and just comparing people with HIV to people without HIV would risk a misclassification bias, namely people with low exposure to HIV who remain HIV negative would be classified as resistant to infection when in reality they just had not been fully exposed. Um, and by the time we were doing these studies, it was clear that there were a variety of epidemiologic factors that modify the risk of HIV acquisition. There were clearly factors associated with condom use versus unprotected sex, with condom use protecting and unprotected sex increasing risk of acquisition. If the partner living with HIV was on antiretroviral therapy, that reduced the risk of acquisition, whereas if they had no antiretroviral therapy and had increased levels of plasma HIV that increased the risk of HIV acquisition. Male partners who were circumcised had reduced risk versus the male exposed partner being uncircumcised, increased risk of acquisition. And there were other biological factors such as pregnancy and sexually transmitted infection status that also modified the risk of HIV. Um, but those factors, as biological factors, were added further complexity, whereas we wanted to develop an approach that captured the epidemiologic exposure that people um, could have that modify their risk of acquisition so that we could take that out of the picture and account for that before we look for other biological factors. So the different couples study design allows for a unique opportunity to quantify exposure factors from both partners, since you can identify frequency of unprotected sex, level of plasma HIV, and whether the exposed male partners were circumcised. You can identify those at the enrollment visit, and you can build statistical regression models that quantify the longitudinal association of these factors with HIV acquisition risk and generate an HIV risk score that takes all the individual factors for a given person and aggregates those to an estimate of an exposure risk score that captures all that epidemiologic information and allows you to quantify that. So that means that we could take, once we quantified the HIV exposure, we could look at the people at the extreme level of HIV exposure, people highly HIV exposed, and after following them longitudinally, identify which of those individuals remained HIV uninfected after long-term HIV exposure, despite high HIV exposure risk. And similarly, we could match by age, gender, and ethnicity to partners who did seroconvert to HIV positive. Similarly, taking the comparable group with similar levels of HIV exposure and identifying those who seroconverted at lower HIV exposure risk. And putting these two groups together, we could develop an extreme HIV acquisition phenotype design where we were comparing people with extreme epidemiologic risk, namely both at very high exposure, um, where the outcome is the opposite of their risk profile, namely that the people who remained HIV uninfected at high exposure risk versus people who did seroconvert at lower exposure risk. And this study design has the high likelihood of identifying genetic factors which have a high impact on disease risk. So we put that regression analysis together and developed an exposure score within two of the cohorts, one clinical trial and one observational study of heterosexual couples. And that exposure risk score, as one would predict, correlated with HIV risk, where the higher the exposure risk, the more likely a person was going to acquire HIV. Um, and we were able to identify, as I just described, 50 HIV exposed partners who remained HIV uninfected despite persistently high HIV exposure. And so therefore we avoided the misclassification of including individuals who had low exposure and remained HIV uninfected. These were 50 people who had high exposure who remained HIV uninfected, and then matched by age, gender, and ethnicity to 50 individuals who were HIV exposed, who HIV seroconverted at lower HIV exposure. And our study plan for this genomic study was to perform a discovery analysis by doing complete whole genome sequencing on these 100 individuals and evaluate for genome-wide associations with HIV acquisition risk, and then to do a replication analysis to retest the strongest of the associations from that discovery analysis in a second cohort of highly HIV exposed partners. So the next step in this study was to ask the question, what genomic variation should we assess and how do we analyze it? The issue there is based on the fact that most genetic studies of HIV and of many other genetic factors at that time, this is in the early 2000s, had focused on common single nucleotide variation that's relatively common. That means greater than 10% frequency in the population. And that was based on the idea that common variation was likely the type of variation that's associated with disease, while also recognizing that analysis of less frequent variants has much lower power. The fact that a variant is less frequent in the population makes it less likely, lower power that you can actually detect an association with disease. Um, but in the last couple decades of research, we've identified that low frequency and rare variants have the propensity to have strong effects on disease risk. And so we wanted to focus on this low frequency, recognizing this, we wanted to focus on low frequency functional variants. Um, and we took the approach of saying we would increase our power to detect associations of these low frequency variants by aggregating their variants with similar characteristics in what's called a variant burden analysis. And to just give a brief description of that, the variant burden analysis, these are techniques called RBT1 or SCAT-O. There are multiple techniques now to take this approach. Group variants with similar characteristics, namely by gene, by exon, or by gene region, and try to assess the association of those variants with an outcome. So in an example here, a hypothetical example, we have a gene with four variants marked by the 1, 2, 3, and 4 in this gene region. Um, if partner A has variants 1, 2, and 3, and partner B has variants 2 and 4, and partner C has no variants in this gene region, then partner A would get a score of 3, a burden score of 3. Partner B would get a burden score of 2, and partner C would get a burden score of 0. And then we could take that burden score and test whether it's associated with HIV acquisition to test whether variants in this gene or gene region are meaningfully associated with HIV acquisition. So the first part of our study was a discovery analysis looking at the gene with the strongest association for variation burden with HIV acquisition risk. And we found that that gene with that strongest association with risk was CD101, where we found that for each additional variant that CD101 has in an individual, it was associated with a more than 2.5-fold increased odds of acquiring HIV. And that was nearly statistically significant, but didn't reach genome-wide statistical significance. And so that meant that we wanted to do this replication study. But to do the replication study to test it in a second cohort, we needed to identify a subset of variants to look at. So let's take a look at the gene structure and the association with variants of CD101. CD101 is the gene located on chromosome 1, and um, it has a variety of protein features. As I mentioned, it's a transmembrane protein with the extracellular portion of the protein including a variety of immunoglobulin-like domains, 7 in total, as well as a transmembrane helical domain and a short cytoplasmic domain. Um, each of these regions of the protein were associated with variants, and in particular, there were five variants in the immunoglobulin-like domains that were associated with, in the discovery analysis, were associated with HIV acquisition risk. There were four variants in the cytoplasmic domain, and there were two variants in the untranslated regions, the three prime untranslated regions. So this gave us a set of variants within this gene that in the discovery analysis had some level of association with HIV acquisition. And now we could take this subset of variants and test them specifically in a replication group, a second group of individuals from one of the clinical trial studies that we had conducted, um, where we did targeted sequencing of CD101 in 261 highly exposed participants, 53 of whom had acquired HIV. And what we found when we divided these variants that we genotyped into the different categories, the IG-like extracellular variants, the cytoplasmic variants, and the 3' untranslated region variants, we found that the strongest association, this hazard of fourfold increased risk, was associated with statistically significant association with the IG-like variants. The cytoplasmic variants had a lower hazard that was not, when you, after accounting for multiple comparisons, was not statistically significant, or barely non-significant. And the untranslated regions did not have significance either. Um, and the interesting part was that although we had looked specifically at low frequency variants, the fact that these were multiple variants that we were aggregating together meant that 20 to 25% of Africans retained at least one or more of these IG-like variants. So although low frequency, the aggregate burden analysis meant that this had actually significant impact or implications um, for susceptibility to HIV in African populations. We did a second analysis or second component to this replication analysis to look at individuals with lower exposure to HIV. And that analysis, when you looked at the or calculated a protected sex index, that's the proportion of visits where the female partner reported use of a condom during vaginal sex. What you can see by this graph is that as you decrease the protected sex index, which means you increase the exposure to HIV, you have a steady increase in the hazard rate of any IG-like variants. And so what this suggested is that um, the two interpretations you could have to this data is that HIV infection depends on HIV exposure, which we knew in the past, and that would be kind of a no-brainer interpretation, or that components of HIV exposure modify the inflammatory potential of CD101 variants. That is, as you get higher levels of exposure, there's a gene-environment interaction where the CD101 variants are actually causing more inflammation or more likelihood of infection than individuals who have low exposure. So I want you to hold this thought in your mind, and we'll come back to this later in the presentation. Now that we've identified CD101 as being the gene with the greatest risk associated for HIV associated with variants, what was known about CD101 at the time? It was identified as a transmembrane protein regulating T cell activation in 1997, and was found to be expressed on lymphocytes, monocytes, and antigen-presenting cells. And a number of studies during the early 2000s in mouse models had demonstrated that in a graft versus host disease model, that regulatory T cells, Tregs, with an increased level of the reference CD101 expression were more effective at suppressing activated T cells. And variants in CD101 Ig-like domains, immunoglobulin-like domains, were associated with type 1 diabetes in mice, and um, knockouts of CD101 in the germline of mice resulted in increased levels of mucosal Th17 cells and increased colonic inflammation, colitis. Um, so the conclusions from this suggested that increased CD101 expression would be inflammation inhibiting or homeostatic, but reduced levels or expression of a dysfunctional CD101 could have pro-inflammatory effects. So the question is, what's the phenotype that's associated with CD101 Ig-like variants? And that led to our second collaboration with Dr. Jenny Lund's lab at the Fred Hutchinson Cancer Center here in Seattle. And in this context, we focused on a descriptive and functional characteristics of circulating immune cells, the peripheral blood mononuclear cells, PBMCs, that have been cryopreserved from partners in the cohorts that we had been looking at. Um, and we were able to look at partners with versus without CD101 variants from the fact that we had genotyped a variety of individuals and had their cryopreserved samples that we could now look back at based on their genotype. So, how do CD101 Ig-like variants impact the distribution of cell subsets was our first question. And we used cryopreserved PBMCs from HIV exposed people living without HIV to compare immune cell subsets for those with versus without variants in the Ig-like domains. And we found differences in the frequencies of T and B cells, monocytes, and dendritic cells. But I'm going to highlight the specific impact of CD101 variants on subsets of regulatory T cells. So this figure, this whisker plot, shows that on the top box, it shows comparison of different subsets of regulatory T cells among people with no variants in the circles and people with any Ig-like variants in the boxes. And you can see that there's strong statistical significance in the difference of CD101 positive regulatory T cells in individuals being increased in individuals with Ig-like variants compared to individuals with no variants. And similarly, there are for other subsets of CD101 positive regulatory T cells, there were similar associations. And we found to a lesser extent, one of the cytoplasmic variants was also associated with increased levels of regulatory T cells with CD101 expression. Keep this in mind again. We'll come back to this later in the presentation of CD101 positive Tregs in cryopreserved PBMCs. But this showed that the level of these CD101 Tregs were increased in partners with CD101 variants. But then the question is, how do these variants affect the regulatory T cell functions? It's harder to assess regulatory T cell functions because in general practice, regulatory T cells suppress inflamed or activated T cells. And so looking at that functional effect of suppression is harder to do. But Dr. Lund's lab took the approach of doing an indirect assessment comparing whole PBMCs versus PBMCs where regulatory T cells have been depleted. And in those two comparisons, stimulate with a common viral antigen, something that most people have been exposed to, Epstein-Barr virus in this case, and look at use the proportion of CD4 T cells that are expressing IL-2 as a measure of reflecting the Treg function, with the idea that as you deplete Tregs, you should see an increase. If those Tregs were functional, you should see an increase in the level of activated T cells, which are no longer being suppressed by functional regulatory T cells. So, for example, in this plot here, which shows compares whole PBMCs versus Treg depleted PBMCs, looking at the proportion of activated CD4 T cells, you can see that for individuals with no variant, where their regulatory T cells were functioning normally, when you take away regulatory T cells, in general, you increase the level of these activated T cells, IL-2 positive T cells, in the mix. And when you have whole PBMCs with functional Tregs, in general, that level of activated T cells comes down because of the regulatory T cell function. Well, when we did that experiment with IG-like variants and cytoplasmic variants, we found that IG-like variants showed a significant um, get my pointer up here, showed a significant reduction in regulatory T cell function. So as you see, the Treg depleted did not change as dramatically from the whole PBMCs, because the Tregs in the case of IG-like variants were less functional, and therefore you get less of a change in the activation of CD4 T cells by removing Tregs. And there was less, although appearing like cytoplasmic variants had a similar effect, it was not as statistically significant in this third panel. And you could also look at just the level of IL-2 positive CD4 T cells and see that IG-like variants were associated with a lower level of the IL-2 um, CD4, IL-2 positive CD4 T cells than no variants. So, so Tregs with CD101 IG-like variants are less functional at suppressing T cell activation. We also looked at CD4 and CD8 T cells and looked at whether the IG-like variants modified their ability to express cytokines. And we found that in both CD4 and CD8 T cells, when you use the Epstein-Barr virus stimulation, you found that the IG-like variants in both cases showed a statistically significant increased level of the interferon gamma cytokines. And so from that, we concluded that CD4 and CD8 T cells with CD101 variants have increased potential to make cytokines. And finally, we also looked at gene expression in CD4 and CD8 T cells, and we found that this plot here shows the either the upregulation in red or downregulation in blue of particular genes in individuals with variants, IG-like variants, compared to those without IG-like variants, and the statistical significance of that change um, in expression is on the y-axis. I'm having troubles with that pointer here. Um, so on the y-axis, the statistical significance of this fold change in expression. And so you can see from here that CD4 T cells with CD101 Ig-like variants may be intrinsically more susceptible to HIV because they're expressing, um, um, sorry, they're expressing HIV restriction genes, MX1, ISG15, IRF7, BS2. These are all um, HIV restriction genes that restrict or target HIV in the cell. These are all expressed at lower levels in individuals um, with the variants compared to those people without variants. So CD4 T cells may be more susceptible because they are less expressing less of these restrictive genes. And similarly, CD8 T cells in this similar plot, they you can see um, that they downregulate granzyme B, which is a gene that's involved in cytotoxicity for killer T cells, cytotoxic T cells to kill cells infected with viruses. CD8 T cells with CD101 Ig-like variants may be intrinsically less effective at cytotoxic responses. Um, but these, as I described, those are all in um, peripheral mononuclear blood cells, circulating blood cells. We didn't have samples from mucosal compartment to see how CD101 is affecting immunity in the portal of HIV entry within heterosexual sex. And so we collaborated with both Dr. Lund as well as Nelly Mugo, who works in Thika, Kenya, doing clinical trials and observational studies among participants in Kenya. We worked with the two of them to collaborate on a study that looked at CD101 variants in samples from genital mucosal biopsies, um, where we did an observational study of HIV serodifferent and seroconcordant negative heterosexual Kenyan couples, 406 couples, following them longitudinally um, through initiation of PrEP and ART and evaluations for STI and BV. And we collected samples of whole blood for DNA as well as for PBMCs. And then we also collected cervicovaginal fluid and cryopreserved vaginal and cervical biopsies with the idea of testing for the effective Ig-like variants on these genital mucosal and circulating cells. And the first thing we've identified is that the Ig-like variants are associated with altered levels of cytokines, of soluble mediators, cytokines and chemokines in the genital mucosa. This is a list of mediators that had statistically significant changes associated with the presence of Ig-like variants in the genital mucosa expression of these mediators. And as you can see that amongst in general, these factors were all reduced in the genital mucosa of individuals with Ig-like variants. And two of these have the RANTES and the leukocyte inhibitory factor are both factors that have been shown to have increased levels known to protect against HIV infection. And so reduction of these mediators would be associated with increased risk of HIV infection, which is what we're seeing with the Ig-like variants. In addition, we used these samples to answer the question of whether there's evidence of cellular changes in the genital mucosa with CD101 variants. And among individuals, and this is with biopsies from the ectocervix vaginal mucosa, as well as comparing to PBMCs, we did multi-parameter flow cytometry looking at T cell characteristics of these biopsies or these tissue samples, and found that any individuals with any Ig-like variant had regulatory T cells with increased levels of or detectable levels of CD101 in the vaginal tissues that were higher um, in the IG-like variants compared to no variants, and that was statistically significant. Um, we also saw that Th17 cells, inflammatory cells in the mucosa, um, were expressing CD101 at higher levels in the vaginal mucosa. This didn't reach statistical significance, but when we changed to looking at people with only IG-like variants, people with any IG-like variant meant that it was they had an IG-like variant, but could have had other variants as well in CD101. Only IG-like variants is a more specific category where they only had IG-like variants without any other variants in CD101. And what we found was we replicated the CD101 positive Tregs association um, again, but we also found that the CD1 positive Th17 cells were now statistically significant with a stronger association in IG-like variants. So, so again, if you remember, I asked you to keep in mind, remember that in the circulating cells study, we had found that CD101 positive Tregs were at higher levels in with IG-like variants, individuals with the IG-like variants compared to those who didn't have IG-like variants. And now we're seeing that this is replicated and, if anything, enhanced, as well as showing that there's also enhanced Th17 cells, which are inflammatory cells, now all in the vaginal mucosa, which is where the first encounter with HIV on a sexual exposure is going to happen. So these all sort of lend further credence, these data lend further credence to the association of this variance, these variants with increased risk of HIV. The last collaboration here I'm going to talk about looks at studies of the impact of variation on innate immune responses, trying to look at cellular mechanisms by which CD101 variation might impact the likelihood of HIV acquisition risk. And this is in collaboration with Dr. Florian Hladek's lab at the University of Washington and his lab team members. And just to back up for a second here, innate immune responses that we're going to look at here are associated with looking at cells that recognize pathogen-associated molecular patterns, that is, pathogen molecules that are detected from the cell surface and other structural molecules of pathogens or nucleic acid modifications. And these responses are hardwired, but they have rapid responses in hours to days, and they're not highly specific, but can at least get the immune system responding to pathogen exposures. And in terms of intracellular responses, those tend to be more targeted to antiviral responses. Toll-like receptors tend to be one component of this innate immune response, where toll-like receptors there's 9 toll-like receptors in humans, and numbers 1, 2, 6, 4, and 5 are all cell surface receptors that respond to bacterial agents or bacterial pathogen factors. And then TLR 3, 7, and 9 it's 3, 7, 8, and 9 are factors that are receptors in this intracellular compartment that respond to viruses and intracellular bacteria. And these receptors, the toll-like receptors, initiate the innate antiviral response through activation of antiviral type 1 and 3 interferons, release of cytokines, and then eventually activation of the adaptive immune response. And stimulation with toll-like receptors elicits a wide range of pro-inflammatory mediators. And from this table, you can see that stimulation of toll-like receptors 1, 2, 6, 4, 5, 8, and 9 are all associated with eliciting inflammatory cytokines like TNF alpha, IL-1 beta, and IL-6. Um, so Dr. Hladek's team looked at what the effect on CD101 expression was, RNA expression was, when you stimulated with different TLR agonists, and found that um, with stimulation of TLR 4, 5, and 8, there was a downregulation of CD101 transcription in healthy donor PBMCs. These we because TLR8 is so focused on viral responses, we focused on better understanding this relationship of TLR8 stimulation with CD101 downregulation in through subsequent experiments. Um, and in this study, what we did was used PBMCs from people with CD101 variants and compared that with PBMCs from people without CD101 variants, stimulated both sets of PBMCs with TLR8, and assessed the level of CD101 expression after 6 hours of TLR8 stimulation versus after 24 hours in people without variants versus people with variants. And we found that at 6 hours, there's clearly a reduction in CD101 expression in both, but with a stronger reduction in people with variants. And this persisted and actually got stronger in people with variants at 24 hours, such that that was a statistically significant change that is associated with variants having a higher and a longer duration reduction in CD101 in response to TLR8 stimulation. So, further studies by Dr. Hladek's team showed that it was predominantly in antigen-presenting cells and to a lesser extent in regulatory T cells that this CD101 downregulation occurred. Interestingly, if the regulatory T cells were sorted away from the other cells in PBMCs um, prior to stimulation, you lost that effect. So it's quite likely that the regulatory CT cells are not directly responding to TLR8 stimulation, but the regulatory T cells are responding to something else from some other cell type. So it's an indirect effect with regulatory T cells. But we also found that inflammatory responses, IL-6, IFIT-1, and interferon beta, all increased in proportion to the level of CD101 downregulation. Um, so that's consistent with the idea that CD101 inhibits inflammatory responses so that as you downregulate CD101, inflammatory responses increase. Um, and um, and the fact that that happens more in people with variants indicate that there's more pro-inflammatory mediators that are present in people with variants, and that would enhance the likelihood of HIV acquisition in cells within people with variants. CD101 has been recently highlighted for other reasons. It's been shown to be a marker for tissue-resident memory cells. It's been shown to play a role in exhausted effector cells in chronic viral infection. And interestingly enough, it's been found that CD101 positive Tregs are prominent targets for HIV and SIV, simian immunodeficiency virus, and during chronic ART may contribute to the latent reservoir of HIV. And again, think back to the studies that both in the mucosa as well as the PBMCs, where we found that CD101 Ig-like variants were associated with increased levels of CD101 positive regulatory T cells in the vaginal mucosa and PBMCs. So this again goes along with the idea that this could be a source for increased levels of HIV acquisition. Um, I want to focus on one last paper that was published last year where a group looked at mice and did gene editing techniques to remove CD101 from specific cell types within the mouse, and then challenged them with either a chemical inflammation challenge or a bacterial infection challenge. And in both cases, it was found that CD101 expression in regulatory T cells was necessary for control of gut inflammation. But they also found that CD101 expression in myeloid cells, specifically neutrophils, was necessary for the control of Salmonella bacterial load through the production of an innate antibacterial metabolite. So what this says is that CD101 function differs by cell type and encompasses both innate anti-inflammatory and innate antibacterial responses. Um, so before I tie up the presentation, I just wanted to touch on one other gene disease association that we identified in addition to CD101 that is of very great interest. And that is that we identified AXL as being the gene with the strongest association between the presence of variants and protection against HIV. CD101 had a strong association with risk of HIV. AXL has a strong association with variants mediating protection against HIV. AXL is one of three receptor tyrosine kinases expressed on antigen-presenting cells, and less is known about its role, but some evidence points to it limiting the immune response duration and intensity, so tamping down the level of inflammation. Um, and the intriguing part is that Ramel McElprang has recently looked at the cohorts that we had been following and demonstrated that um, if you separated people by the number of CD101 versus AXL variants, the line in blue shows individuals who had AXL variants only, and this is a survival curve of probability of remaining HIV negative. Um, and so people with AXL variants only had the highest probability of remaining HIV negative. People with CD101 variants only had the lowest probability, had the highest probability of seroconverting to HIV positive. Um, and people without any AXL or CD101 variants, or with both AXL CD101 variants, showed an intermediate level of susceptibility to HIV. So, what this suggests is that among highly HIV exposed partners, the ratio of CD101 to AXL variants may help determine a risk of HIV acquisition. So, finally, to tie up, I would summarize by saying that variants in CD101 are associated with increased risk of heterosexually acquired HIV, and that the mechanisms may include homeostatic function of regulatory T cells, leading to increased levels of activated T cells in circulation, as well as CD101 positive regulatory T cells and CD101 positive Th17 cells in the genital mucosal tissues, reduced expression of HIV restriction genes in CD4 positive T cells and cytotoxicity genes in CD8 positive T cells, and reduced level of genital mucosal cytokines inhibiting HIV, as well as an accentuated reduction in CD101 expression and increase in pro-inflammatory mediators in response to innate immune activation through TLR8. Just to tie up here, the question is, should we be barking up another tree? Um, Much of the last 2 centuries of vaccinology is focused on eliciting adaptive immune responses to protect against infection. Our improved understanding of CD101 and AXL may help provide ways to enhance innate resistance to viral and bacterial infections. And this could have implications beyond HIV, having a role in response to diverse viral infections, a role in response to diverse bacterial pathogens, and a role in host commensal relationships. So maybe we need to be barking up a different tree. Um, and so finally, to conclude the long road to translation interventions, we're following up on these things with ongoing studies, looking at the generalizability of this to other cohorts with regards to CD101 and AXL, and documenting the effect of CD101 and AXL in mucosal inflammation models, and then also working with Niari at the ICGEB to look at the role of CD101 Ig-like variants in HIV susceptibility and HIV reservoir in South African men. And clearly long-term challenges, including needing to identify the natural or artificial ligands that bind CD101 and AXL. How do they actually translate their capacity to respond into molecular changes? And to identify key points in pathways for CD101 variants that impact HIV risk and identify drugs that could alter those effects. And this is my colleague Connie Kellums in Kisumu, Kenya, saying "Never say die." And as has been attributed to Nelson Mandela, it always seems impossible until it's done. So we just keep plugging on. And I need to say thanks to all the collaborators that produced all these data and all the people involved in the studies that we recruited cohorts for, the Partners in Prevention study, the Couples Observational study, Partners Prep study, and the Kinga study team. And I think and then clearly to thank you to all the dedicated study participants, site staff, and community and other stakeholders. And I think that's all I have.